Yeah, so I'll just give a brief introduction again.
Okay.
Dr. Hua received his master's degree in 2005 and his doctorates in 2009 in biomedical engineering and electrical engineering at Johns Hopkins University. His doctoral training centered on the development of novel MRI technologies for in vivo physiological imaging of the brain, such as protein content and CBV. After completing a postdoctoral fellowship in radiology, he became a faculty member in the Department of Radiology at Johns Hopkins and the Kennedy Krieger Institute. So today we are going to hear about differential laminar activation that dissociates encoding and retrieval in the human medial and lateral entorhinal cortex. Thank you and welcome, Jun.
Thank you so much, Tyler, and thank you for the organizers to have me. Okay, let me go to next slide. Okay, yeah. So as Tyler said, I work at the FM Kirby Research Center at Johns Hopkins and Kennedy Krieger. So by training, I'm a MR physicist. So I do methods development, of course, and application in brain diseases, mainly functional MRI and physiological MRI, blood vessel, and CSF imaging, etc. So the work I'm going to present today is in collaboration with Dr. Arnold Becker from the psychiatry department at Johns Hopkins. So Arnold, by training, he's a neuroscientist, but he also had a very rigorous training in neuroimaging. So it's been a great collaborate for many years. So the goal of our project is that we want to see, because Arnold has been doing research in MCI dementia, in the ADRC, center at Johns Hopkins. So the particular interest in the memory system. So we want to see if we can apply the laminar function MRI methods to look at, particular component in the memory system. So for years, they've been looking at, the hippocampus region and also the entorhinal cortex region. They've been looking at using especially structural imaging and the 3D functional imaging to look at the, changes in aging and the dementia in these regions. So in particular, the entorhinal cortex, based on my understanding, of course, from Arnold's insights. So the entorhinal cortex has 6 layers. The so-called superficial layers is more associated with so-called encoding of information, or considered the input component to the pathway to the hippocampus. And the deep layers are more associated with retrieval of information, or considered output layers. So there have been lots of studies, of course, in the literature from animals, and as well as some more recent study from humans, subjects to look at functional activities in these 2 different group of layers. So for our study, we're particularly interested is to see if we can use laminar functional MRI to tease out the functional activation or functional response to encoding versus retrieval in these 2 different group of layers. If we could do that, comparing to conventional 3D functional MRI they've been doing, they're looking at the activation, sort of ensemble activity from both group of layers. If we do that, we can separate the encoding and the retrieval responses. That could be an important step towards the application of these methods to aging and dementia study in the memory system. So that's our hypothesis. So we set off, of course, first we need to see the structure. So this is approximately where the entorhinal cortex is. And my understanding is that usually has this wedge shape, but for some other people, it can have 2 turns as well. So very complicated and then strange shape to deal with. And normally you can separate it as as the medium and lateral entorhinal cortex, which has a slightly different function in terms of information processing. So what we did, we did these manual segmentation with Arnold's group on a high resolution T2 image to get these, these structures. That's the first step. And of course, and because of this, a shape and a special location, it's actually very challenging to do functional MRI in this region. One is the irregular shape, so we had to do manual segmentation. And then we have to do isotropic voxels, otherwise it's going to be difficult to tease out the layers. And the second is probably, you can guess it, as an MR physicist, this is very close to the cavity. So there's when you do high resolution EPI, you often see this so-called dropout distortion. This is actually mentioned in one of the recent publications, and they say, because of that, especially the lateral part, it's closer to the cavity, so it's very difficult to have a robust function MR signal. And even the medial part, based on our observation, because the bottom is very close to CSF, the cavity as well, so there's some problem as well. So we tried our best on 70. We want close to whole brain acquisition. We tried of course the conventional EPI method, spin echo as well. But as you can tell, there's still some dropouts. When we do whole brain, it's very difficult to optimize that. So we eventually used this so-called T2 prepared both functional MRI. This is just a simple method we developed a few years ago. It's not something particularly new, and there are other methods, similar methods as well. So basically, the idea is you have a so-called spin preparation module, where you can generate the both contrast, and then following that, you can do a very quick fast GRE similar to MP-Ridge type of readout, which has a much wider bandwidth and much less susceptible to these dropout artifacts. So we did that. As you can see, these are the so-called T2 prep both images. The contrast is slightly different because of the T2 contrast, and also very fast 2-second TR readout for whole brain. But the thing is, if you look at these regions, you can actually get a very better signal from these dropout regions. And also, if you look at the shape of the brain, it's more similar to the structural imaging because the readouts are similar. So this gives us some confidence that when we do segmentation, especially for small regions, the distortion and misalignment could be less of a problem. So we like I said, originally we developed this method to image some kids when they come to our studies. They have metal, dental braces, and then this is even our 3T. You can see, even with a metal dental metallic brace, you see a big hole in the brain. But with the method, we can actually get robust signal. So, and then we thought, okay, so maybe we should use this method for this particular region as well, although it's a T2 based contrast. So we tried that. So this is approximately where we want to image. And, with GRE, you see some, funny signal here, but with T2 prep, it seems that we're getting more robust signal. This is the entire volume we got from this. So eventually we decided to use that particular protocol. We were able to get to 0.9. We couldn't achieve 0.8, but we saw this close enough. And then we got a 3-second TR with partial brain coverage for this particular region. So this is the imaging part. So what's most important that we thought, from our data is, when we look at the temporal signal to noise ratio across the layers. So if we use the EPI, if you look at the EPI data, you see this sort of gradient, of temporal signal to noise ratio across layers. And also you saw a difference between medial and the lateral entorhinal cortex. But if you look at the T2 prep data, it seems that the temporal signal to noise ratio profile is much more uniform across the layers. So we thought eventually we decided to use this method for this particular project. Okay, since I'm short on time, I'll skip the next slide, and unless there are physicists in the audience wants to discuss that. So this is the imaging part. Next, so this is the task we designed. This is really from years of work from Arnold's group, because we want to look at encoding versus retrieval of information. So this is a memory task they've been using. So basically, the subject is presented with a picture every 3 seconds. So there are 3 different types of pictures. So these are so-called repeated pictures. They are basically the same picture, just present repeatedly. These are called similar pictures. They are both apples, but slightly different. And the rest are so-called foils. So they are completely, excuse me, unrelated. So the subject, basically, every time they see a picture, they're supposed to press a button. They tell us whether this is similar, old, or new. That's the task. So for what's the contrast for encoding and retrieval? So for a repeated pair, if the subject correctly identified the second presentation as a old or repeated image, so the first trial of this image is considered successful encoding, and similarly for the similar pairs, and of course the second trial is considered successful retrieval. So that's the task. Okay, so we did this in a few young and bright graduate students who can stay in the scanner for a long time to test our results. And this is really a very long task. It's about 40 minutes to get enough trials, and we separate into eight runs. And we have structural imaging, and these are the analysis with Renzo's help. So this is really the main results from this study. So this is the laminar profile from this is CSF boundary. This is Y matter. This is the superficial layer and the deep layer. So the contrast is encoding versus retrieval. So successful encoding trials versus retrieval trials. So so we see there's a specific peak corresponding to the encoding condition in the superficial layers in both the medial and the lateral entorhinal cortex. And it seems the lateral is response is slightly stronger. And then we see similar retrieval response. It's not negative, it's just the contrast. So retrieval response in the deeper layers in both media and the lateral entorhinal cortex. Okay, so so I'm gonna skip the next few slides, but I can provide a quick summary. So basically, this is the main contrast, and then after that we'll look at a few additional contrasts that basically we want to look at. For example, encoding versus foil. We use foil as a baseline, and then retrieval versus foil as well. So the main finding is it seems that the repeated trial and a similar trial, in terms of encoding, they're very similar. We didn't see much difference in terms of signal response, but in terms of retrieval, they're very different, and we can see signal response difference, especially in the lateral, in the deep layers. So we can detect that. Okay, okay. So to summarize, basically, we think we're happy. We think we have established a protocol that seems to be able to detect a layer-specific response in deep and superficial layers in the entorhinal cortex. And of course, the goal is to apply this in different diseases. For example, based on Arnold's insights, for example, in Alzheimer's disease, it seems the superficial layer is more affected, while in dementia in Parkinson's diseases, it seems that the deep layer are the target to look at. So we're in the processing of looking at these regions. So the reason I want to skip a few slides is I actually have a few additional slides. If I have 1 more minute, I want to show some more recent work from my group. to look at the olfactory region. So we've been we have lots of interesting olfactory region, and we've been doing olfactory function MRI for several years now. So I thought in the spirit of a layer specific MRI. So as you know, of course, the olfactory region is very small and difficult to image. In particular, the most difficult but the most important part, the region is the so-called the olfactory bulb region. That's the first region in the brain in the olfactory pathway. So we want to do, we thought if there's any hope, we can do layer-specific functional MRI in the olfactory bulb, but the first thing is we need to see it, right? So we found this very interesting study almost 10 years ago. So this is actually a specimen. They put a 9.4 Tesla and they image it for 60 minutes. They can actually see layer structure very clearly in the olfactory bulb, but that's 60 minutes. So they did it on 3T as well, 100 minutes, and they can see something. If they do 6 minutes, they can see something, but probably not very clear. So we start to try that as well. And this is actually from last year. So now we have higher resolution. This is this is a 9-minute T2 scan we perform on 7 Tesla. So olfactory bulb is here. This is the olfactory bulb, and not the bright part. The bright part is CSF. That's the thing underneath. That's the olfactory bulb. And usually people view this in coronal view, but when you do axial view, you can see, I think we start to see some sort of a layer-like structure in the olfactory bulb. If you look at the, cross section, we can see some signal peaks. So, of course, we're very excited, but on the other hand, we should be very cautious, to make sure this is not some artifacts or some other structures. They're actually layers. I don't know yet, but I think in the spirit of a layer function, layer MRI, I'd like to show these results and to share with everybody. Okay, I'll stop here. Thank you again.